Thank you, Dr. Gagi and the organizing committee for the kind invitation and the opportunity to showcase our products. Hello, everyone. I'm Vanati. Since June 2021, affiliated to Curtin University and Sir Charles Gardner Hospital as an internationally recognized research expert with 27 years of research excellency in Perth and India in a PhD CEO and principal medical oncology scientist role, founded Medong Therapeutics business model in Perth, Western Australia for viable circulating tumor cell and spheres cancer blood diagnostics in early cancer models. We are way to cancer liquid biopsy clinical trials for radiotherapy treatment. Our goal is to reduce healthcare cancer cost, burden, drug toxicity, and invasive procedures. With this introduction today, at this meeting, I will be talking about novel secreted frizzle-related protein 4 oral cancer liquid biopsy. Globally, cancer is a significant health challenge with 19.3 million new cancer cases and 9.6 million mortality in all demographic populations. People older than 45 have an increased risk of oral cancer due to poor oral hygiene, and lack of dental care and not following regular oral hygiene practices may cause an increased risk of oral cavity cancer. In today's presentation, I will be focusing on the biomarkers as liquid biopsy diagnostic testing to improve the treatment challenges. So now let's talk about oral cancer, otherwise called as mouth cancer. Oral cavity, including lips, gums, tongue, cheeks, and lining of the mouth, when these cells mutate, it becomes cancer. The risk factors are lifestyle, genetic, and external environment factors, and pathological conditions such as Epstein-Barr virus, EBV, and human papilloma virus, HPV, which are sexually transmitted disease, and tobacco use of smoking cigars, pipes, snuffs, and chewing, and heavy alcohol use. So the symptoms are sore or lump, white or red patch in the gum, tongue, tonsils, or lining of the mouth that doesn't heal for a long period, which accounts for 40% of all malignancy in parts of India and Southeast Asia. In Australia, in 2021, it was estimated that about 151,000 Australians will be diagnosed with cancer at the rate of 413 per day, and 49,000 will die 135 per day. So the overall survival of oral cavity squamous cell carcinoma is stable at 50% for very long time, which has not changed for 40 years with present treatments. So we need to introduce advanced diagnostic testings and treatments. We need to act in early diagnosis and treatment and introduce metastasis prevention and screening approaches for a quality of life. Liquid biopsy for cancer care. What is liquid biopsy? A liquid biopsy is an assay that is obtained from body fluid, alternative to surgical complicated procedures. Several body fluids have been shown to have genetic material derived from tumors such as urine, blood, saliva, tears, sweat, and cerebrospinal fluid. The main advantage of liquid biopsies are low risk and minimally invasive. Recently, the liquid biopsy concept has been extended to circulating cell-free tumor DNA. And circulating tumor cells are a rare subset of cells that have escaped from the primary tumor and entered the bloodstream to form metastasis, deposits, or even re-establish themselves in the primary site of the cancer. That's why it's very important to look at viable CTCs compared to previous technologies. And now we are convinced that the analysis of the CTCs and ctDNA are different analytical opportunities, and therefore provide complementary informations, and the approaches is completely different. They are two different analytical informations. The capture of viable CTCs from millions of blood cells is certainly more time consuming than the harvest of cell-free circulating DNA from blood plasma or serum samples. However, functional analysis of whole tumor cells offer the possibility to identify the biological properties of metastatic cells through in-depth in vitro and in vivo analysis, which opens new avenues for basic and translational research that are far beyond sequencing of fragmented DNA present in the blood plasma. So the functional studies cannot be done on the genomics approach. Of course, the genomics approaches will shed some light to get some information for new therapeutics. These cells are more aggressive and accumulate gene alterations by somatic mutations that are the same or even higher than the primary tumor because of additional features acquired in the circulation. The potential application of CTC for clinical use is to acquire a liquid biopsy by taking a reliable, minimally invasive venous blood sample for cell genotyping during treatment to monitor the decline in CTC detectably mutational changes in response to resistance and sensitivity. The prognostic value of CTC in cancer management and personalized medicine for cancer patients requires a deeper understanding at the cellular level, along with other advanced tissue and imaging modalities and technologies. Using a blood test rather than relying on tissue would be a breakthrough, as sometimes a surgical biopsy doesn't take enough tissue or provide the whole body tumor information. We introduced new STEM CTC and sphere technology that got the potential to isolate CTC, showing stem cell-like properties that may possess low therapeutic response or resistance and tumor recurrence. As I said in the previous slide, STEM CTCs, that is CTCs showing stem cell-like properties, may possess low therapeutic response or resistance and tumor recurrence. Wnt signaling plays an important role in therapy resistance. The role of therapy resistance and the Wnt signaling mechanism is not much known in many of the cancers. Circulating tumor cells and its associated circulating cancer stem cells subpopulation and its associated genetic material will be a valuable clinical tool to improve the current treatment approaches for this disease. Developing a less invasive CTC and circulating cancer stem cells as a prognostic and predictive marker will be an added advantage in clinical practice for sequential therapy assessment. In a heterogeneous population of cells in circulations, very few, depending on the genetic makeup, can be cultured successfully for real-time assessment of anti-cancer treatment. So, this opens up further research questions. So, can we identify and count circulating tumor cells in blood samples of cancer patients? The answer is yes, because we were able to establish circulating tumor cells and circulating cancer stem cell spheres in blood samples. Are there specific genetic signatures on these CTCs to indicate therapy resistance and sensitivity? Yes, we will be focusing on CTC Wnt signaling. Is there a variation in CTC concentration during treatment? The answer is yes again. And could we plate these CTCs on ex vivo models for future analysis, that is for drug clinical trials? The answer is yes again. The previous pioneers have established xenograft models. Currently, very little has been published on CTCs and circulating cancer stem cells, heterogeneity and tumor heterogeneity. The primary tumor information and metastasis at different sites within the same patient may have different genomic characteristics due to tumor heterogeneity. Therefore, tumor biopsy alone, which is currently used in clinical practice, may not reveal sufficient information for therapy response and decision making. Combining ctDNA and cell-free DNA genetic information in oncology can complement the current available technologies for future sensitizing therapeutic interventions, mainly for patients who are in end of life stage, in extending their survival without compromising quality of life. CTC culturing for functional studies from cancer patients is logistically challenging. Functional analysis involves understanding the viable CTC metastatic initiating potential using in vitro and in vivo models. The major reason for treatment failure is the anti-cancer therapy modality is due to presence of cancer stem cells, which are more resistant and highly tumorigenic with self-renewing capacity. CTCs expressing stem cell markers are known as circulating cancer stem cell in the circulation. The commonly used circulating cancer stem cells markers like CD133, CD144, CD54 and ALDH are associated with poor patient outcome. As a next step, we are aiming to introduce secreted frizzled-related protein 4, that is SFRP4 CTCs, and circulating cancer stem cell sphere systems for treatment response. Now let's talk about SFRP4 research history since 1994. So what we know and don't know about this molecule. Previous SFRP4 as a biomarker has shown the usefulness for human health as a contraception in reproduction biology, diabetes, cardiovascular disease, and in various cancers. SFRP4 has shown effectiveness in vitro cell culture cancer models, cancer stem cells, and animal models. However, as a new therapeutic regimen, the clinical attempts in human blood circulating tumor cells and tumor spheres as a diagnostic tool are not studied to prolong cancer survival effectively. Recently, as a next step, we can establish these models on circulating cells and epithelial buccal cells, which are organ specific as spheres, will open new effective diagnostic and market potential therapeutic avenues. This slide summarizes the SFRP4 protein history. 20 years of SFRP4 protein history. Highly aggressive cancers are characterized by rapid progression and late metastasis and poor prognosis. There is a need for much improved diagnostics and novel therapies for cancer through detailed biological understanding of the disease. Since 1994, our laboratory has been involved with research into the Wnt signaling antagonist SFRP4. We have previously shown anti-proliferative and pro-apoptotic roles for SFRP4 during normal homeostasis in tissues such as corpus luteum, ovary, mammary gland, and placenta. In 2010, we identified SFRP4 as a potent and anti-angiogenic protein. Our latest study expanded upon this previous work by exploring the effect of cystin-rich domain, the CRD, and the neutron-like domain, NLD, of SFRP4 on endothelial cell activity and cancer mesothelioma cells. What do we need to do for technology improvement in the clinical space? That is for SFRP4 Wnt signaling. So we need to introduce CTC SFRP4 Wnt signaling. Only very few skillful labs can culture viable CTCs for drug discovery in the world. Most labs focused on CTC detection without large patient cohort statistical significance, which limits the commercialization prospects. At Medong Therapeutics follow-up to our previous discovery, we aim to introduce the new generation CTC spheres SFRP4 Wnt signaling treatments to patients. Previous studies have shown SFRP4 are downregulated in mesothelioma tissue and cell lines. In addition, we demonstrated promoter methylation of SFRP4 in glioblastoma models. Our laboratory has previously shown downregulation of SFRP4 in mesothelioma cell lines. We have also investigated vesicular trafficking of SFRP4 and observed that the neutral-like domain has a vesicular association signal. Most significantly, intracellular redistribution of SFRP4 was observed in response to Wnt3a, suggesting Wnt3a can modulate intracellular localization and secretion of SFRP4. Chitosan nanoparticle gene delivery of SFRP4 and its domain gave enhanced biological effects which are characterized by the formation of membrane-associated vesicles, morphological changes. Our results demonstrated the effectiveness of chitosan, SFRP4 and associated domains, the CRD and NLD nanoparticle-based gene delivery, and showing promise as an effective alternative non-viral gene delivery system. So this is my PhD story, which are summarized. All my publications are summarized in this slide. This is a schematic representation of my PhD story. These are my publications for SFRP4 drug discoveries and the related patent. Based on the previous research publications and training, the new title of today's talk on novel SFRP4 oral cancer liquid biopsy was designed. As shown in this slide, in continuation to my PhD finding, we collaborated with Oxford University to study the function of SFRP4 domains and mutant proteins on breast, ovarian, prostate, glioblastoma cancer stem cells. Out of those models, glioblastoma model showed significant effect on NLD treatment. Now let's talk some of the CTC advantage over ctDNA in this slide. The advantage of CTC technologies are otherwise stem circulating cells possess the ability of forming self-renewal and proliferation, producing downstream progenitor cells and cancer cells that drive tumor growth. However, functional analysis of the whole tumor cells offer the possibility to identify the biological properties of metastatic cells through in-depth in vitro and in vivo analysis, which opens new avenues for basic and translational research that are far beyond sequencing of fragmented DNA present in blood plasma. So the viable cells can be useful for cell prediction, treatment prediction, mutation genomics, drug screening, toxicity test, and drug response outside the patients, and also it is easy for clinical application. Now, how the previous research is going to be useful for CTCs. For example, I have come up with a question: are we treating cancers free of CTCs? So that's the question I've come up with. And to answer that, the other CTC lab, Professor Nicholas Esito's lab, already published in breast cancer, which is more exciting for advanced cancer treatments. So these are the open questions for oncologists now. I've put up these questions for us to improve the current treatments. So are we treating and checking for CTCs in the blood? How do we address the current treatment failure? How do we monitor viable CTC for anti-cancer therapy recurrence and sensitivity? Did we test current and the upcoming anti-cancer drugs for CTCs in the blood before treating patients? How do we genetically modify the cancer cells in the blood for effective therapies? In order to address previous question and tackle recurrence challenges, we have introduced STEM CTCs and Spheres technology as a new diagnostic path technologies. In applying Professor Nicholas Esito's FDA drug screening approach to Win signaling, we introduced new improved STEM CTC and Spheres breakthroughs to SFRP4 drug treatments for cancer risk volunteers as incidental discovery. These breakthroughs are in collaboration with Sir Charles Gardner Hospital and Curtin University. We started this research in 2017 and established the technology in 2019. Now, in this pandemic, founded as Medong Therapeutics in translating the discoveries to clinic. And this is the recent patent pending on rapid isolation of stem circulating tumor cells and spheres for cancer prognosis and diagnosis. And this is the previous related review we published. To address the previous questions, Medong Therapeutics have come up with blood testing packages, which is all-in-one stem-like circulating tumor cell. Like we have decided to offer as a standard, premium and elite packages, such as standard CTC counts, CTC line counts and CTC sphere counts, and cancer risk line CTC line counts, and new cytotoxic cancer risk sphere dissociation treatment prescriptions. So the products and services offered for these are the products and services offered for changing people's life. These are the advanced technologies we have introduced recently to advance SFRP4 drug development. The present invention relates to live cancer initiating cells in the blood as compact blood cancer diagnostic testing packages. Being genuinely healthy at the cellular level requires a whole body blood approach looking at the behavior of individual cells. In day to day activities, lifestyle, and demographics, the inner circulation strength is measured, quantitatively measured by the stemness of CTCs and sphere morphologies. It provides a solution to count viable cancer cells and the proliferative cells as spheres in the blood for therapy prediction with an additional benefit of drug testing outside the patients. So this reflects the current cancer status of patients' resistance and sensitivity to treatments. This slide is an overview of the available product as stem potential CTC, spheres, and cancer risk sphere dissociation drug concentration prescription. As shown in the figures, surprisingly, during validation as incidental discovery, 5 volunteers out of 13 negative control groups yielded 6 to 10 days of viable CTC lines. On cryopreservation revival, the viable CTC lines were like solid tumor cancer associated fibroblast as shown in these figures. The proof of concept study was on 22 to 48 years old healthy volunteers group detected viable CTC lines as 38.5% for risk and 61.5% free from cancer. In supporting this finding, recent very small embryonic stem cell OCFO group from India and Singapore with good statistical significance published in Stem Cell Reviews and Reports on known early cancer groups. Our products are unique with drug testing solutions. Live cell immunostaining and genomics comparing to known cancer tissue organ signals, measuring the tumorigenicity on appropriate animal models for identifying the cancer type will open doors to new interesting early cancer detection and prevention drug therapeutics. We require more organ cell type or inflammatory marker signals experimental data. Measures for early cancer screening and follow-up care for cancer risk care patients like our National Institute of Integrative Medicine NIM group should be taken into consideration. In addition to the previous data, we generated additional simple step CTC sphere culturing. We generated late circulating cancer stem cell spheres with site population as well as 1 month high purity spheres with site population. The negative control did not yield any spheres as shown in the figures here. We also followed a conventional density gradient method with site population cancer spheres. And we were able to generate spheres within 18 hours turnaround time with site populations with high purity as shown in these figures. The methods were also simplified to multi-step CTC enrichment, able to cryopreserve and revive viable solid tumor morphology cancer-associated fibroblast like CTC lines. Additionally, we were able to generate numerous primary and secondary spheres from the same wells. Now, with established circulating cancer stem cell spheres, with faster turnaround time and with high purity, we can accelerate CTC sphere drug discovery for Wnt inhibitors. And these are the recent advancement of SFRP4 research. The future of SFRP4 research as a new generation diagnostic tool model are the recent advancement in this field for culturing patient CTC spheres, CTC lines, and proteins. This slide summarizes the first human CTC Wnt inhibitor cluster dissociation discovery. To offer a solution to current treatment challenges, we introduced SFRP4 treatment in short-term CTC for complete elimination of circulating cancer stem cells and cancer relapse approaches. New dose and cell model innovation is introduced, unique to my own previous published SFRP4 concentration on equivalent spheres with limited CTC numbers. Successfully, we generated numerous primary and secondary tumor spheres from spiked CTC lines for SFRP4 Wnt targeted preventive drug screening, alternatives, sensitization, and anti-cancer therapy follow-up approaches. As expected, SFRP4 dissociated the cancer risk aggregates. In this slide, let's see about CTC cell drug development. So CTC can be used for overcoming radiotherapy resistance and sensitivity, and also for advancing SFRP4 CTC drug development clinical trials. Not only that, we can also come up with new drug development platforms, combinations for cancer patient health management. And we can also introduce PET-CT imaging by introducing circulating CTC and circulating cancer stem cell technology. For future global clinical trials with a research track in India and Australia, we are aiming to expand collaboration, engaging national and international investigators. The feasibility study will allow us to overcome radiotherapy resistance and introduce a sensitization approach, advancing SFRP4 drug development and introducing new drug development combinations for including naturals for cancer patient health management. We could also improve imaging modalities, introducing CTCs and circulating cancer stem cells specific biomarker detection in the targeted regions such as SFRP4. In the future, I will be concentrating on SFRP4 liquid biopsy in the cancer diagnostic space in collaboration with global leaders, and looking forward to learning about more drug development clinical trials with other global mentors. To address more questions about the role of SFRP4 in liquid biopsy, we will introduce CTC drug development, effective CTC sphere, dissociation drug concentration for effective treatment prescription, and predicting treatment resistance and sensitivity for targeted therapies. And we can also use for manufacturing native SFRP4 CTC protein production. We can also use this technology for stratifying patients, generating saliva SFRP4 cancer IDs for upcoming Wnt inhibitor treatment selection. This slide is more about introducing native SFRP4 CTC protein medicine, and this will be a new addition to CTC technology. Towards future research direction for producing native patient SFRP4 proteins using their own CTC or circulating cells and sphere cells for all diseases than our conventional recombinant bacterial cloning methods will be a quality product, faster and effective for human healthcare applications. The advantages of native protein are it will be E. coli and animal free, and the origin will be from human blood and saliva, and it can be purified using non-denaturing methodologies, properly folded, and fully it will be functional, and it will be biologically active for patients than our recombinant protein because it is from E. coli origin, and it will be a stable formulation to retain their functional activity and direct human SFRP4 domains, and CRD and NLD can be produced, and this can be used to treat all disease models outside the patient before treatment. I'm thankful to my CTC research group at Curtin University and at Sir Charles Gardner Hospital for the seed funding and the clinical trial training to